Welcome to Click for IB Biology. In this last of D3.3 homeostasis, what we're looking at is a comparative analysis of the plasma, filtrate and urine produced in the osmoregulation of the kidney. There's a PDF of this presentation that's linked in down in the description. Download that, annotate that with your own notes, and then why not take the 10-question quiz, check the answers, so that you've got your understanding of this part of the course. And so what I've done is I've generated a comparative table. You can see here that there's the substance. This is column here is the range of the substance found in the plasma. So that's pre-ultrafiltration. Then here we have the glomerular filtrate. So that's post-ultrafiltration. And then the urine will be after selective reabsorption. So if we annotate that here, so this is ultrafiltration, that may help you to remember. So this is after ultrafiltration, and this is after selective reabsorption has occurred. So this is the actual urine content. So water, glucose, amino acids, urea and uric acids, these two largely reflect the excretion of nitrogen components, whereas these three are more associated with osmoregulation. And so if we look at the plasma, what we find is that in the glomerular filtrate, most of the water has been filtered out. But then, as we already know from our study of the kidney, although 80% of the water is reabsorbed in the proximal convoluted tubule, and then even perhaps another 10 or 15% in the loop of Henle, it's actually the collecting tubule which does the fine adjustment of the water there. And of course, that depends on the secretion of ADH under the conditions of hydration or dehydration. If we look at glucose, glucose is circulating in the plasma at this level. It's completely filtered out, so we got complete filtration into the filtrate here. But notice that then we go to zero, which means that we have no glucose in the urine because of full selective reabsorption in the proximal convoluted tubule. And indeed, if one does find glucose in one's urine, that is an early indicator of the condition known as diabetes mellitus, diabetes as studied under other topics on this course. Amino acids, again, follow a similar pattern to glucose, where we generally find that there are no amino acids in our urine, again, fully reabsorbed in the proximal convoluted tubule, and then used in the synthesis of proteins. Now then we look at this curious combination here. So there's a full filtration to this point, but then we have a concentration of the urea and also a concentration of uric acid present in the urine. And this is because of the removal of water as it is selectively reabsorbed in the proximal convoluted tubule, the loop of Henle, and the collecting tubule. And so this leaves the urea with less water in which to be dissolved, and so therefore more concentrated. And of course, this is consistent with the idea of the excretion of these nitrogen-based molecules. So one might anticipate a similar type of data present in the IB examination. You can apply your theory of the kidney to resolving these differences. That brings us to the end of our study of D3.3 homeostasis. There's a link there to the channel, so you can subscribe. That is massively appreciated. And of course, there's a playlist as ever for your convenience on all of this particular topic. Thank you for listening.